Hi students, and welcome to chapter 5 dealing with the cell membrane. In this chapter we're going to look at the structure of the membrane as well as osmosis and diffusion, facilitated diffusion, as well as the responsibilities of the membrane. So to give a little history, at the turn of the century, researchers noted lipid-soluble molecules entered cells more rapidly than water-soluble molecules, suggesting lipids are a component of plasma membrane. Later, chemical analysis revealed the membrane contains phospholipids. Now, in 1925, Gorter and Grendel, they found an amount of phospholipid extracted from a red blood cell was just enough to form one bilayer, suggested that nonpolar tails directed inward and the polar heads directed outwards. Now, to account for permeability of the membrane to non-lipid substances, Danieli and Davison proposed sandwich model, which was later wrong, in which it said that the phospholipid bilayer, that the sandwich had a phospholipid bilayer between the layers of protein. Now, with electron microscope available, Robertson proposed proteins were embedded in outer membranes, and all membranes in cells had similar composition of a 3/4, the unit membrane model. Now, in 1972, Singer and Nicholson, they introduced the currently accepted fluid mosaic model of the membrane structure. The plasma membrane is a phospholipid bilayer in which protein molecules are partially or wholly embedded. Now, the embedded proteins are scattered throughout the membrane in irregular pattern, and it varies among the membranes. Now, electron micrographs of freeze fracture membranes supports this fluid mosaic model. Now, the fluid mosaic model, which we see here in this diagram, it has two components. It has lipids and it has proteins. Now, the lipids are arranged in a bilayer. Most plasma membrane lipids are phospholipids, which spontaneously arrange themselves in a bilayer. Now, the nonpolar tails are hydrophobic and they are directed inward. The polar heads, now these polar heads, they are hydrophilic and are directed outward, which are here. And they're going to face the extracellular fluid and the intracellular fluid. Now, the plasma membrane also contains glycolipids, which you see right in here. These glycolipids with the structure is similar to the phospholipids, except the hydrophilic head is a variety of sugars. Now, they are protective and they assist in a variety of functions. We also have a cholesterol, and the cholesterol is a lipid found in the animal plasma membrane, and they're there to reduce the permeability of the membrane. Now, at body temperature, the phospholipid bilayer has consistency of olive oil. The greater the concentration of unsaturated fatty acids residues, the more fluid the bilayer would have. Now, in each monolayer, the fatty acid tails, they're going to wiggle, and the entire phospholipid molecules can move sideways at a rate of about 2 micros, the length of a prokaryotic cell per second. However, the phospholipid molecules, they rarely flip-flop from one side to the other. The fluidity of the phospholipid bilayer allows cells to be pliable. Now, what does pliable mean? Pliable basically means it can be easily bent or it's flexible. Now, some membrane proteins are held in place by our cytoskeleton filaments, which we see here. And most drift in the fluid bilayer. Now, the transmembrane protein, well, I'm jumping ahead of myself, so let's move on to the next slide. And we're going to look at a detailed photo or diagram of this phospholipid structure. We see that it has a nonpolar hydrophobic tail and has a polar hydrophilic head. And you notice we have three types of views of this same phospholipid. Here we have the formula, the chemical formula, and we have the space filling model. Then you have the icon. When we look at the formula in A, the pink area would be your glycerol. The yellow area is your phosphate group. And this green area, I'll get it going here, this green area here is that charged choline, which we see here at the top. Now when we look at the cellular membrane, it has four compartments. It has your phospholipid bilayer, which we briefly talked about. That's flexible, has a flexible matrix, and its barrier is permeable. That means it will allow things in. When we look at the transmembrane protein, it is a major component of every membrane, and is going to extend through both sides of the membrane. They have a hydrophobic region that's embedded within the membrane and a hydrophilic region that will project from both sides of the bilayer. Now, many transmembrane proteins are glycoproteins with a carbohydrate chain that projects externally. Now, some are anchored to the membrane by covalently attached lipid or covalently bonded to the carbohydrate chain of a glycoprotein. Now, others are held in place by a non-covalent interactions and are disrupted by gently shaking or even a change in the pH. Now, their function basically is to help with transport and communication across the membrane. Now, many integral membrane proteins are not fixed in place, and they can move about just as the phospholipid molecules do. The third component is the interior protein network. Molecules are structurally supported by intracellular proteins that reinforce the membrane shape. For example, a red blood cell has a characteristic biconcave shape because a scaffold made of a protein called spectrin. It will link these proteins that are embedded in the plasma membrane bilayer with the actin filaments in the cell's cytoskeleton. Membranes use networks of other proteins to control the lateral movements of some key proteins within the bilayer, anchoring them to specific sites. Our fourth component is the cell surface markers. Membrane sections are assembled in the ER, which is our endoplasmic reticulum. Then they're going to be transferred to the Golgi apparatus, and then from there taken to the plasma membrane. Now, within that passage, what happens during that passage, the ER, the endoplasmic reticulum, it adds change of sugar molecules to the membrane proteins and lipids, converting them into this the glycoproteins and the glycolipids. Now, different cell types exhibit different varieties of glycoproteins and glycolipids on their surfaces, which act as cell identity markers. Here we have a table, which is also found in your textbook on page 97, and it gives you the breakdown of the four components I just went over: the phospholipid bilayer, the transmembrane proteins, interior protein network, and our cell surface markers. Here they are. Here are the compositions of each. We have the function of each, as well as how it works. Then we have an example of each. You will see the functions again, as well as how it works. Just let you guys know. And maybe a few of these compositions. So make sure you guys study this chart in its entirety so you'll be able to answer the correct questions on your exam. Here we have another diagram that's in your textbook on page 98, and it gives us a microscopic view of the plasma membrane, and is using a freeze factor microscopy. Here we have that the cell was frozen, and then they used a knife blade to crack it open. Now, here the cell often fractures through the interior. So you see that yes, they did cut it, but you notice part of the cell was fractured, and a little piece of it is in the box that was removed. Now, you'll see that we'll have the hydrophobic area of the bilipid, the lipid bilayer is going to split it's actually not going to split, but it's splitting the membrane into two layers. And when we get a closer look at the layers, you see that the plasma membrane is separated so that you're able to see the second half of the lipid bilayer. You'll be able to view the tails, the hydrophobic tails. And here is the fractured upper half of the bilayer. In this diagram here, it says the exposed membrane is coated with platinum, which forms a replica of the membrane. The underlying membrane is dissolved away, and the replica is then viewed with electron microscopy. So when we take a closer look at our phospholipids, we'll look at the structure in details. Well, as we discussed in an earlier chapter, we noticed that the phospholipids have heads and they have tails. I also mentioned it in the previous slide, or 2 slides back. These phospholipid heads, they are hydrophilic, meaning that they love water, and they are polar. The tails, which we have here on either side, are hydrophobic, and they do not like water. So they're considered to be non-polar. So what happens when we put a molecule, or a phospholipid molecule, yes, inside of water, the water, let's check out my diagram here. The water, as we know, is oxygen, H2O. So you have one oxygen, you have 2 molecules. So these hydrogens are trying to find something to bond with. They have an affinity to bind, meaning they always want to bind with something. And what happens during that time, since these nonpolar tails dislike water so much, they sort of like turn their back away because they don't want to be touched. It's just like a cat. A cat hates water. That's what it's going to do. It's going to run away. It'll scamper out of the water as fast as it can to get away from it. And that's the same thing with these tails. They're going to turn away from it, positioning the polar hydrophilic heads towards the water molecule, causing the hydrogen bonds to bond with the heads. And in that turning, it will form these two layers of tails facing each other, and none of the tails will come into contact with the water. And this is what forms this phospholipid bilayer. Now, this layer is formed very spontaneously and is driven by the tendency of water molecules to form the maximum amount of hydrogen bonds. Now, when we look at this lipid bilayer, it's stable because of water's affinity to hydrogen bonding never stops. Basically, they always want to bond, like I said in the previous slide. These hydrogen molecules will always want to bond with something. Now, the hydrogen bonding of water, it holds the membrane together, which is great. Now, although the water continually drives the phospholipid molecules into this bilayer configuration, it does not have any effect, on the mobility of the phospholipids, relative to their lipid and non-lipid members, in the bilayer. So they're not going to affect the movement of what's going on with the two layers, the phospholipid, the hydrophilic and the hydrophobic areas. Now, because the phospholipids interaction is relatively weak with one another, the individual phospholipids they might become unanchored. They have these unanchored proteins and are comparatively free to move around within the membrane.